Welcome to join the webinar. Today, we are going to talk about the automatic pipetting system for qPCR sample preparation. This is Fred Zhu speaking. During the epidemic of COVID-19, the qPCR system, nucleic acid extraction systems are all in great demand. However, you still need the automatic system for the sample preparation to save your time and to avoid human error. We, Blu-ray Biotech, have qPCR system TurboQ, and also we have automatic system EasyMate to help the sample preparation of COVID-19 detection. So next, we will give you an introduction of EasyMate, and then share with you an applications of COVID-19 detections. Let's welcome Sonia to talk about it.
Hello everyone, I'm Sonia and I'm the product specialist of EasyMate. So today I will talk about the topic about EasyMate's automated liquid handling system for COVID-19 applications. So let's go first about an introduction. So what is EasyMate? EasyMate is an automated high-precision pipetting system for molecular biology experiments. So the design concept of EasyMate is to replace the tedious and repetitive pipetting traditionally, which is performed by handheld manual pipetter. So this means that EasyMate is a machine that can save hand-on time for the lab and can reduce human error. And more, it's an easy-to-use machine. On the EasyMate, there's an automated pipetting module, which is also called APM. It is interchangeable and no tools are required to change it. And the software to operate the EasyMate is very easy to learn. You can learn it and master it in 1 hour. Last but not least, EasyMate has high accuracy and precision. The lowest accurate and precision is down to 1 microliter. And now EasyMate have 4 kind of system in our series. We have EasyMate 401, 601, 401S, and 601S. And I will tell about the introduce all the function in detail in next slide. So this is the EasyMate system layout. If you order an EasyMate, you have an old EasyMate system in your laboratory. You will have an EasyMate platform, this one, and you will also have a computer which have easy starter software to operate the EasyMate. The EasyMate platform has a functional safety door, and you need to have interchangeable APM on it, which has 1, 4, and 8 channel, and we offer for 2 kind of volumes. And here you have to put adapters on the platform. Then later you can put your labwares on the adapters. So the 401 system, the most basic of EasyMate, will look like this. On the work table, we have a disposable usage tray, which use the tip, and this one is it's called 2 reagent areas that we have 2 areas to let the user to put kind of adapters called reagent adapters. And here, we have 4 SBS format area, which is a little bigger than the reagent areas to put the labwares on it. So the 601, a little complicated kind of system than 401. It means the 601 means it had 6 SBS format area. So the work table will be in like same logic, the tip tray, 2 reagent area, and SBS format area. The 401S and 601S, the difference of them between the 401 and the 601, the basic one, is that they have an additional HEPA module and an additional UV module. if user need more to have a temperature control for the solution, we can offer the user to have an optional active cooling and heating module. And this is the place to set the module on it. Okay, so we have a form that on the work table, user have to put adapters on here for like 2 reagent areas for for the most of 2 reagent adapters, and SBS area for the most of 6 SBS format adapters. And now I'm going to talk about what is called adapters. So adapter is the things that we put on the work table and adjust to the labware. Like you can see, if the users, they want to use PCR plate, like 96 well PCR plates, or strip tube, or deep well plate, just those kind of labware that use a lot in the laboratory, they need to put it on the adapter, then to put it on the work table. We also, on the reagent areas, we also have the specific adapters for the tube and bottle or strip tube. And if the user, they want to use the large amounts of solution, we have a kind of adapter that is called reservoir adapters that offer the user for large amounts, like 80 milliliter and 15 milliliter. So to put on the SBS format areas, we also have tube adapters, like we have 20 well adapters or 48 well adapters of tube adapters to put tube on the SBS format areas. Okay, so today let's start today's main topic, how to apply EasyMate on COVID-19. So now, now every lab is focusing on COVID-19 pandemic. So about EasyMate, how EasyMate apply on COVID-19 tests is we can use EasyMate to do the qPCR preparation. Like we can prepare master mix, you can transfer master mix, you can transfer the sample and the control by EasyMate. Then you can just send your plate to run the qPCR. So I'll offer that you for like 3 examples. Now we already have users, how they use EasyMate in the COVID-19 applications. So this is the first example. The user, they have an EasyMate 601 and a 4-channel APM. And the materials to put on it, they need to have their own kits, and they use the microcentrifuge tube and the PCR plate. So this is so they use the EasyMate like this. They use it on transfer the master mix and transfer the sample, transfer the control, and then they run qPCR. And because then you can see the total time is less than 15 minutes per 1 set. So this is the equipment. Okay. So this is how we first, how the procedure first go. They transfer the master mix first. First, they put on the 4 tube of master mix on the work table. And here they will put an empty 96 well PCR plate here and the tips here. Although here now it's empty, but this is actually the sample and control place. So first, the transfer master mix will look like this. The APM will pick master mix from the reagent area. They will add it to the empty, now it's empty qPCR PCR plates, and we'll keep going on to fill all the plates. So this plate will become, after this transfer master mix procedure, it will become a 96 well PCR plate, but now with master mix. Then later, the procedure is continued with the transfer sample and control procedure. So you can see here is 94 sample tubes and 2 control tubes. So the APM will start from this place and add their sample to the PCR plate. They pick another column add it to the PCR plate, and keep continue till the end. They pick the sample with the control and add it to the PCR plate, and also the positive control. So you can see here, because this is a 48, one of this adapter is 48 well. So if you put 2 kind of it, it is 96 well as total. And then a 96 well of tube equals to a 96 well of PCR plate. Then you can just put this PCR plate into the qPCR machine. So this kind of set is less than 15 minutes. Okay. And this one is another example. Then user use the 601 and 401. Okay. So they use they prepare the they transfer the master mix, they transfer the sample, the control, and then they run the qPCR. Now this is the equipment. So you can see the little difference is here. The first example, they use the 48 well kind of adapters, and this they use a deep well plate adapter. So this is their work table. They put the master mix here. Here are the four deep well plate that is they take from the extraction machine, and here is the 96 well plate, and here they put the tips. They will pick the master mix to add it to this PCR plate, and this is the transfer master mix procedure. So first it's empty, and then later they will be full of PCR, full of master mix. Then transfer the sample and control. Here is the deep well plate, and then this is how they set the samples and the control. You will see this kind of, the blue will be the samples, and then the purple one is the negative and the positive control. They pick it from the four channel APM pick from here, and then they add it to here. And in less than 10 minutes, they will finish this a set of 48 well of PCR, half a set of PCR plate. No. And the third example, they use a 401 system with a 1-channel APM. The set they use EasyMate on more steps. They prepare master mix on here. They transfer the master mix, the sample, and control. And because they use a 1-channel APM, this is a longer process that need around 45 minutes. Okay, so this is the equipment. Okay. Continue so it looked like this. Okay, first they prepare the master mix from the super mix and the enzyme mix into this kind of master mix plate. Then later, they will transfer the master mix from this two well into the 40 well of PCR tube. They use the tube, not the PCR plate. And the last, they transfer from this 48 tubes of samples into the strip tubes. Okay. So you can see that EasyMate, now we have 3 examples of how the user applied it on COVID-19 application. And they all use different kits and different libraries from different companies. So it means that if you are more interested about the application, it doesn't. This 3 example is just an example. You can contact Blu-ray for more information about, if you already have a kind of kits that you already use it and you have your own libraries, and then you can send us an email and we can tell you more about how you can just apply it on EasyMate. Okay, so now let's go a little more about EasyMate. So let's quickly recap the features of it. We talked about it in the introduction. So it is an easy to afford automated pipetting machine. It is very easy to use, and it's easy to service. That the calibration can be set the mail-in APM. You don't have to send the whole EasyMate for the calibration, and it's accurate and precise. Okay. So like here, more application is for the users. This is from the general pipetting test to the nucleic acid purification, cell culture, or more like genomics application, where very complex assays or kind of in vitro diagnosis. This all kind of application we already have users that use worldwide. And because there are more applications, that means they have different labwares. Different labwares need different adapters. Here you can see this is some kind of special adapters. That is like this one is for HLA typing. This one is a kind of special adapters. It's for only for the PCR Max Echo system, and it is a 48-well qPCR plate. And also we have users that they want to use their own labwares, so they definitely need to customize. And Blu-ray can offer users to have customized adapters. You can see the three kind of it. This is all real examples from our users. And also, this one is a kind of special adapters that is for a kind of economic way for the users. If they use kind of temperature sensitive solution that need to keep in cool surroundings, but they need to build an economical way, they can try cool block adapters. That cool block adapters is an adapter just to preface in the micro 20 degrees Celsius for like 2 to 3 hours, and it can maintain under 7 degrees Celsius, like this port, for around like at least 1 hour. And still, if the user need very kind of precise temperature control, we have active cooling adapters to put on the EasyMate that really can control the surroundings to be around like that 37 degrees Celsius or 50 degrees Celsius. That is based on the users, their requirement. Okay. So this is some reference sites that worldwide already have users that using EasyMate. We have like Seeing Bioscience, which is a company that do NGS, and then it's based in Taiwan. And in the Health Research Institutes in Taiwan, or the hospital in Taiwan also have our EasyMate. And still more like Turkey, South Africa, Israel, and FDA and the USA have our EasyMate. So our EasyMate is definitely trustable. So this is just a short webinar of our EasyMate. If you have more questions, you want to know more about it, please please let your comment in the Q&A, and we will send you back. Okay. Now we have a question about what kind of tips is it used. To use an automated pipetting machine, you need the machine kind of tips that is only for automated pipetting machine. And our tips is you can use the Beckman tips or oxygen tips on EasyMate. If you want to know more information, we will send you the email later. Okay. Okay. So okay. Now we have someone raise hand. Can you leave your question in the Q&A? Okay. So both single. We have another question that can we have both single channel and multi-channel pipette coexist on the system? I'm sorry that if you want to use 2 APM, then you need 2 machines. You cannot use both only on one. Okay. So is there more question, or I will give the comment back to Fred.
Okay, thank you, Sonia. I believe you have many questions about this webinar. So please leave your question. We will reply you by your registered email. Okay. And also after this webinar, we will send you the video and also reply your questions. So now we answer 2 questions. Can we make out the single channel and load the multi-channel on the same head? You can use one machine and you can change the APM manually. So in fact, if you want to, if the procedure you only need one channel APN, you can just use one channel APN. But if you need another protocol, need multiple channel, you can just replace the APN menu, and then you can use the protocol.
And we have one more question: is how easy is it to calibrate the EasyMate and the respect to the depth of the tips that go into tubes? I would say it's very easy. That you can. We have training lessons for the distributors. That distributor can do this calibration by themselves, and it is. I believe that this is as easy that you can master about how to calibrate it in 1 hour. So, using we have another question. Would you recommend using filter tips, particularly for COVID work? That depends on your kits. So you can use filters. Of course, you can use filter tips if you require it. Okay. So, is there any more question? If more, please leave the comments, and we will send how this reply to you again and by mail. Okay, so.
Okay, so we also have many questions. We will reply you by email. And thank you for your attention today. Okay, please get our reply by your requested email. So thank you today. Bye bye.